Hello, students, and welcome to another session of Learning with Funke. And today we're going to be talking about the pentose phosphate pathway. Okay, so what is the pentose phosphate pathway? Why is it important? And what are the reactions that occur in the pentose phosphate pathway? We're going to be going over that today. And so one of the things that we need to know is, we've talked about glycolysis. If you haven't seen that video, please go back and watch the video. But we talked about how in glycolysis, we go from glucose to glucose 6-phosphate, and then that goes all the way down, through glycolytic breakdown steps to produce pyruvate, which, then undergoes further oxidation in the citric acid cycle. However, that isn't the ultimate fate of G6P. G6P can also undergo other fates, oxidation of G6P to pentose phosphates. In the pathway we'll be talking about today, the PPP pathway, the PP pathway, pentose phosphate pathway. It's also known as the phosphogluconate pathway or the exose monophosphate pathway, but for the most part, it's commonly known as the PP pathway. Okay, and so what we see here is that in this PP pathway, we have, glucose 6-phosphate going to 6-phosphogluconate, and then goes to ribulose 5-phosphate, goes to ribose 5-phosphate, and then, the importance of this is this ribose 5-phosphate then goes on to make our nucleotides, coenzymes, DNA, RNA, which is, of course, very essential for all living cells. And, there is also a tandem pathway that goes kind of opposite to that. So while we are going in the oxidative pathway, we are going from glucose 6-phosphate to ribulose 5-phosphate and to ribose 5-phosphate. There's a non-oxidative phase. So we have two phases, right? The oxidative phase and the non-oxidative phase. And the non-oxidative phase actually takes ribulose 5-phosphate via catalyzation with transketolase and transaldolase enzymes and takes that back to glucose 6-phosphate. But we're going to be going over all of that in, and the reactions in the subsequent steps. And so we talked about the first pathway, the oxidative pathway. In the oxidative pathway, it is important to know that here, contrary to glycolysis where we had NAD pluses and NADHs, now we have NADP plus as the electron acceptor. And NADP plus, comes into this pathway where glucose is being converted to converted to 6-phosphogluconate. And in this series of steps, we're getting NADPH. NADP plus gets reduced to NADPH. It can also get regenerated back during glutathione reduction back to NADP plus to continue back through this process, kind of like how we talked about in the glycolysis video, how with fermentation, generating back the electron acceptors that we need to go into the process. Rapidly dividing cells like your skin, your intestinal mucosa, and even tumors, they use this pentose ribose 5-phosphate to make RNA, DNA, and other coenzymes like ATP, NADH, FADH2, and coenzyme A. And in other tissues, the PP pathway, the essential product isn't just the pentoses, but you also need this NADPH as it's needed for reductive biosynthesis, or to it's important to help with countering damaging effects of oxidation radicals in the cells. All right, so now let's go straight into the PPP reactions. I've provided the overall reaction for the pentose phosphate pathway. You pretty much go from glucose 6-phosphate. You use 2 NADP pluses, right, with water, and the end product is ribose 5-phosphate, carbon dioxide, and 2 NADPHs, of course, and 2 hydrogen ions. All right, so for the first step of the oxidative phase, we're focusing on the oxidative phase, and then we'll go to the non-oxidative phase next. Here we see that the first step, we go from G6P, glucose 6-phosphate. This is catalyzed by G6P dehydrogenase, and the first thing we form is 6-phosphoglucono lactone. And this is an intramolecular ester. And here, like I said before, NADP plus is the electron acceptor. And then in the second step, after we formed this 6-phosphogluconolactone, the next step is catalyzed by enzyme lactonase, and that pretty much uses, like the first step, uses magnesium as a precursor. takes in water. Water is one of the reactants, and then we form 6-phosphogluconate here in the second step of the oxidative phase. All right. And now when we move into the third step, now we are going from 6-phosphogluconate and converting that to 6-phosphogluconate, using that with 6-phosphogluconate dehydrogenase as the enzyme, we end up making the first 5-carbon phosphate that we talked about, D-ribulose 5-phosphate. And ribulose 5-phosphate, here we see we're generating a second molecule of NADPH. It's important to know that. And this ribulose 5-phosphate is important in glycolysis regulation and gluconeogenesis as well. And lastly, in the fourth step of the oxidative phase, we go from ribulose 5-phosphate, right? Then we convert it to its isomer. Remember how we talked about isomerization reaction? So we're just kind of like a rearrangement. So we're going from ribulose 5-phosphate, a 5-carbon ring, to another 5-carbon ring, its isomer, which is an aldose, ribose 5-phosphate. And this is catalyzed by phosphopentose isomerase. Keep in mind, you need to remember these enzymes, as these are typically concepts that are tested on exams. Okay. And so here, once again, I've provided the overall reaction for the PPP pathway. All right, so now moving into the next phase, we've talked about the oxidative phase. Now, the non-oxidative phase, this is where we are, pretty much recycling those pentose phosphate and creating glucose 6-phosphate again. In the past slides, we went from glucose 6-phosphate to, ribose 5-phosphate, but here we're going from, those pentose phosphates, like ribose 5-phosphate, and going ribulose 5-phosphate and going to glucose 6-phosphate. And so what we'll see is that tissues that usually require NADPH, the pentose phosphates produced in the oxidative phase of the pathway are typically recycled back to G6P via steps catalyzed by transketolysis and transaldolysis. And so in the first step of the non-oxidative phase, we see here we start with ribulose 5-phosphate catalyzed by ribulose 5-phosphate epimerase, and that gets converted to xylulose 5-phosphate. We can see here it's going from 5 carbons still to 5 carbons, but that's just catalyzed by this epimerase enzyme. And then after this first step, we have a series of rearrangements of the carbon skeletons, and then we end up having 6 5-carbon sugar phosphates converted to 5 6-carbon sugar phosphates. So we have 6 5 carbons, that's 30 carbons in total, and then 5 6, which is 30 carbons. So we're still conserving, mass. The mass is mass balance wise or element balance wise. We still have 30 carbons going to 30 carbons, right? And this completes the cycle and allows for oxidation of glucose 6-phosphate while producing NADPH in the process. And the continual recycling, will lead to ultimate conversion of glucose 6-phosphate to 6 carbon dioxide molecules. All right. So for a better picture-wise context for those who are better visual learners, like I was saying before, you start with, ribulose 5-phosphate here. When we're going this way, we're going the non-oxidative way. We've already talked about the oxidative pathway from going from here to here. But now for the non-oxidative, we're pretty much focusing at the bottom step. So we talked about how we go from ribose 5-phosphate via isomerization reactions. We convert that to xylulose 5-phosphate, right? Some of that go to xylulose 5-phosphate, not all, because you have to have this reaction between both of them. And then ribulose 5-phosphate and xylulose 5-phosphate kind of react with this enzyme, transketolase enzyme, to produce, there's a 2-carbon transfer, right? And then we end up producing pseudoheptulose 7-phosphate, which is a 7-carbon a 7-carbon compound rather. And then we also produce a 3-carbon compound, G3P, right? We talked about this also in glycolysis. So we have 5 carbons plus 5 carbons, gets rearranged, and then we end up having still 2 compounds, but 7 carbons and 3 carbons. After that is formed, then these 2 compounds react again, catalyzed by transaldolysis, and then to make F6P, which is a 6-carbon compound here, and then a 4-carbon compound. So we're going from 7 plus 3 to 6 plus 4, right? Still 10 carbons. but this is catalyzed by a different enzyme, transaldolase. And then after that, this E4P, this carbon compound, then reacts with a cellulose 5-phosphate catalyzed by transketolase again to form fructose 6-phosphate and G3P. So we see here we're going from 4 carbons, 5 carbons down here to 6 plus 3. And something else that is important to note is we see here that we are having kind of like those alternation of enzymes, right? So the second step, we have transketolase, then the third step, transaldolase, then this is the first step. The first step is just isomerase and epimerase. But the second step, transketolase, third step, transaldolase, fourth step, transketolase. And then when you form G3P and F6P. Remember in glycolysis, the video we talked about how you can go from F6P to G3P, but here we're going in the opposite direction, right? Kind of like in gluconeogenesis, but then from G3P, you can have that, catalyzed by this TPI enzyme and the aldolase enzyme in a series of steps, and then finally by fructose 1,6-bisphosphate. So these three enzymes will take you to fructose 6-phosphate. And then, we have fructose 6-phosphate here and here. That can be catalyzed by phosphohexose isomerase. We talked about this also in the glycolysis video, the previous glycolysis video made. That can take you back to glucose 6-phosphate. And these oxidative reactions can keep happening, right, to take you back again to ribose phosphate. So all of these are kind of like happening at the same time. We're having something going here, and there's, a complex, intricate, regulation going on in our systems where, sometimes something has to be down-regulated, sometimes something has to be up-regulated, depending on the cell needs, right? And on the right, the key point to note here is if you count the number of 5 carbons, which pretty much relates to, it's just this shown here with the number of carbons, we end up having 1, 2, 3, 4, 5, 6. So we have 6 5 carbons, right? And then we end up having how many 6 carbons? 1, 2, 3, 4, 5, right? So if we start with a 6 5 carbon process, we end up with a 5 6 carbon process. It's still overall 30 carbons, right? But that's the overall summary of the reaction flow for the non-oxidative phase, rather. All right, so going much deeper, we said in step 2, we have that transketolase enzyme that uses TPP as a cofactor. The cofactor TPP, thiamine pyrophosphate, is needed for stabilization for the formation of two carbon carbanions that form during this process. But pretty much we go from cellulose 5-phosphate in this step to ribose 5-phosphate, and this enzyme transketolase is catalyzing the transfer of carbon 1 and carbon 2 highlighted here of the cellulose 5-phosphate to ribose 5-phosphate to form this 7-carbon compound like I just went over, pseudoeptulose 7-phosphate, and this 3-carbon G3P. So you can see here that this G3P pretty much comes from the cellulose 5-phosphate, right? And then this ribose 5-phosphate is pretty much the ribose 5-phosphate plus the carbon 1 and carbon 2 previously from the cellulose 5-phosphate. All right, so next step, after we have formed, S7P and G3P, these two will also undergo a transaldolase catalyzed reaction. And you can see here, kind of similar to the aldolase reaction from glycolysis that we went over. There's a 3-carbon rearrangement where these 3 carbons are removed from pseudoeptulose 7-phosphate and condensed with G3P to form the rest of this here. So this becomes a 4-carbon compound, E4P, and fructose 6P, which is just this initial 3 carbons plus this added 3 carbons. So we have a 4-carbon compound plus a 6-carbon compound here. All right, so transketolase acts again. Remember how I said they kind of act in alternation, transketolase, transaldolase, and then transketolase again from steps 2 to steps 4. So in step 4, we have transketolase acting again, and then we are going from fructose, pretty much we're forming fructose 6P and G3P from cellulose 5-phosphate and E4P that we were the products of the last step, right? So these products from the last step are pretty much going again and forming G3P and F6P with the cofactor TPP. And so that gets you to here, right? So now we have this F6P and G3P. And like I went over before, then you can have those enzymes catalyzed, in glycolysis, but this time we're going the reverse direction, right? From G3P, and then you go to, using TPI, we go to G3P and DAP. And then from that, pretty much all the way till we get to F6P, catalyzed by these three steps, three catalysts to get to F6P. And then phosphoexoisomerase can catalyze the formation of this. From F6P, we can go to 2G6Ps, right? Because we have two F6Ps here, 2G6Ps. Okay. Now that we've gone over, the oxidative steps and the non-oxidative steps. So pretty much how for the non-oxidative phase, we go from G6P all the way to ribose 5-phosphate, which end up going, into making nucleotides, coenzymes, DNAs, RNAs. We also went over, how to go the reverse direction. So how do we generate, go back to generating glucose 6-phosphate? I like to think of it as glycolysis versus gluconeogenesis, right? So glycolysis going one way, and then gluconeogenesis kind of like the reversible, where you're going in the other direction. So similar to this, we're having in the PPP pathway, we have an oxidative phase that kind of like oxidizes metabolites from G6P to ribose 5-phosphate and to ribose 5-phosphate. And then in the other way, we are having like that non-oxidative phase, or like think of it like now we're reducing. Here we were oxidizing things, now we're reducing. So we are going over like a non-oxidative or reduction phase, and then catalyzed by these two enzymes, transketolase and transaldolase, and we're regenerating G6P again. And in this whole process, in this oxidative phase, we're generating NADPH, right? 2 NADPHs. And this NADPH, for continuation of the process, this NADPHs can, get reduced again during glutathione reduction to form NAD plus to continue this process all over again. But in terms of regulation of NADPH, we have to understand that NADPH plays a very key role in regulating, whether we are doing G6P partitioning between glycolysis, because we talked about in the previous video, G6P can go through glycolysis, to, form pyruvate. However, G6P can also, it's also an important metabolite or compound, if you will, that goes through the PPP pathway. So the availability of NADPH can also determine whether or not G6P is available for the PPP or glycolysis. And so how does that work? So as NADPH formation increases, we have higher NADPH. Maybe we are forming more NADPH compared to how much we're using NADPH. So we know we're forming NADPH as we go through the PPP oxidative phase. So if we're forming a lot of NADPHs, NADPH in your cells will rise, the amount of the NADPH concentration in your cells will rise, and it can go back and inhibit this first enzyme, right, in the PPP pathway. So in that manner, PPP is, inhibited. We don't have PPP going on. Instead, the alternate pathway competing for G6P's attention, which is glycolysis, would happen. And so we'd end up, instead of using G6P through PPP to produce more NADPH, we already have high enough NADPH. So why would you want to keep making more? And so that's the cell's way of regulating how much NADPH we have. And instead, glucose 6-phosphate then goes on through glycolysis to make ATP. And just as an overall summary slide, some key takeaways. We went over the oxidative PPP, bringing about oxidation, decarboxylation of at that first carbon of G6P, and then the consequent reduction of NADP plus to NADPH. And then how that oxidative PPP pathway just reduces NADP plus to NADPH, right? Because when you have an oxidation step, if something is getting oxidized, something has to get reduced. So in the case of, while we're oxidizing G6P all the way down to ribose 5-phosphate, we are reducing 2 NADP pluses to NADPHs, to 2 NADPHs. And the next summary is the first phase of the PPP consists of oxidative reactions that convert G6P to ribulose 5-phosphate and reducing NADP plus to NADPH. The second phase is the non-oxidative steps that convert the ribulose 5-phosphate back to glucose 6-phosphate to begin the cycle again. And next, in the second phase, transketolase, which uses TPP as a cofactor, like we discussed before, and the transidolase, they catalyze the interconversion of, we have 3, 4, 5, 6, and 7 carbon sugars. But overall, what we see is, in these reversible reaction steps, we end up having the conversion of 6 pentose phosphates, so 6 5-carbon compounds to 5 6-carbon compounds, so 5 exose phosphates. And we did that math in the previous slide. If you don't understand, please go back and watch it. But we had that image where we counted the number of 5-carbon compounds and the number of 6-carbon compounds, and we ended up going from 6 5-carbon compounds to 5 6-carbon compounds. All right, and in the carbon acylating reactions of photosynthesis, these same enzymes will catalyze the reverse process. So while we have done this, there's also the reverse process, which is the reductive pentose phosphate pathway, just for your knowledge, and this does the opposite, right? So this goes from converting 5 exose phosphates to 6 pentose phosphates. That hasn't been covered in this class, but just so you know that there's also something called the reductive pentose phosphate pathway that does the reverse of what we just talked about. And lastly, the entry of G6P it can either be used in glycolysis. We have a video on this. If you still don't understand, go back and watch the glycolysis video. Or the G6P can go into using the PPP, into the PPP pathway to make, ribose 5-phosphate, generate NADPH, which has been the focus of this video. And all of that is regulated by the availability of the relative concentrations of NADP plus and NADPH. So that kind of ties into, if we have too much of NADPH, then we stop the PPP pathway, because this can inhibit that first enzyme we talked about. And then instead we just go on to make ATP in glycolysis. And on that note, I hope you've been able to learn the PPP, the pentose phosphate pathway, its oxidative and non-oxidative reactions, and how NADPH is important in regulating the partitioning of G6P into, glycolysis versus the pentose phosphate pathway. I wish you good luck. Once again, if you have any questions regarding this lecture or other topics that you'd like me to cover, please email me at learningwithfunke@gmail.com, and good luck on your exams, tests, or quizzes.